Good evening. Now, this is the financial disclosure, my financial disclosure. So, as you know, Prosigna is a multi-parameter assay, genomic assay for breast cancer. And I am a pathologist, and we start in our department in 2014 to test breast cancer. So, let me explain why a pathologist adopt Prosigna for study breast cancer. First, because it's possible, because it's a localized genomic test, so it's possible to maybe part of the instrument of the modern pathology department. The other point is that it's a simple technique, because it's each target, each mRNA is binding by two different probes. One of that is linked with a fluorescence barcode, and this way is not necessary amplification, and it's possible to study 800 genes simultaneously. This is the assay is made by an hardware, prep station, and digital analyzer, consumable, the Prosigna kits, and a software. A software contains an algorithm that produces the report. This algorithm put together PAM50 genomic profile, clinical or pathological feature, so nodal status and dimension of the disease, and the proliferation score. This is, for this reason, is called second generation molecular assay for breast cancer, because differently by the older one, he put together pathological feature and genomic profile, because the other one used just genomic profile to predict prognosis for breast cancer. So there is another point that there is an analytically validated and decentralized test, and data demonstrated there's a concordance across different sites and different labs is between 90% to 100%. So this is a Prosigna report. It's made by two parts. The upper one is the molecular intrinsic subtype, and the lower one is risk of recurrence. So let me talk a little bit about molecular intrinsic subtype. As Dr. Pratt said in 2009, for the first time, it was demonstrated that with 50 gene, the evaluation of 50 gene is possible to classify breast cancer in 4 different subtypes, molecular subtypes. So luminal A, luminal B, HER2 enriched, and basal-like. They are very different prognostic behavior. For this message, it was so strong that in 2011, the St. Gallen International Expert Consensus Conference decided to adopt this kind of classification to classify breast cancer. Moreover, they indicated, they suggested a therapy and systemic therapy for each kind of cancer. But with time, because since in most of the world may be not available Prosigna test, PAM50 test, because financial or logistic problem issue, it was adopted a surrogate for classification of breast cancer, in immunohistochemistry technique. And so we have triple negative, hormone positive, HER2 positive cancer. This is what the pathologists see under the microscope. So there is a estrogen positive cancer. In this case, it's a positive estrogen positive cancer, like 90% of positivity. Progesterone positive, like 90%. Ki67, 25%, and HER2 probably like 70% of cells are positive. So now, as you know, I use a new term, percentage. Percentage means that the pathologists need a cutoff to decide if this percentage is positive or negative. For this reason, the notes is indicated the percentage of estrogen progesterone, and according guidelines is equal or more than 1%. But there are reports that demonstrated that in case of 1 to 9% of positivity, in the first paper, in the first table, there is just 2 cases on 25 are real luminal. In the different series, in a different group, as you can see, like just 40% of cases with estrogen progesterone between 1 to 9% was confirmed as luminal by Prosigna, by PAM50. The same is for Ki67, as Alex said, and now we have low Ki67, high Ki67, but without any reference about the cutoff for defining low and high. And just what I see under the microscope, this is a breast cancer, this is an H&E section, and this is a consecutive section staining for Ki67. And according to breast cancer working group, a pathologist has to score at least 3 high-power fields at the invasive age of the tumor, and the scoring should involve the counting of at least 500 malignant invasive cells. Means that this is a high-power field, and this is our tumor. Okay. As you can see, there are many hundreds of possible fields that the pathology have to score. And it's possible that one of that, the Ki67, is completely different by the previous one. So the question is, if in this case, this cancer is 90% of Ki67 or 3% of Ki67. So this is the difficult for the issue for the pathologist in the classify breast cancer. And this is a problem. This is a big problem. It's an international problem. There are many work it was made to try to find a reproducibility for Ki67. But according to me, in my opinion, there are no great results about. So for this reason, with Dr. Pratt and Dr. Martin, we put together our series, and we analyzed altogether more than 500 cases scored with Ki67. And the result was that in intermediate group, I prefer the words equivocal Ki67 group, between 11% to 30%, it's very difficult that Ki67 can predict exactly luminal A or luminal B classification. This is the series of my hospital. As you can see in the first graphic, if you consider under 10% to classify tumor as luminal A-like, you're wrong in 18% of case. The same if you consider more than 30% Ki67. There is a mistake in 50% of case. But very interesting is the last graphic in which it's possible to distinguish with the PAM50 the right classification of breast cancer in an equivocal group of Ki67. So let me show you some example of my experience. In this case, I have a breast cancer with estrogen 19%, progesterone 19%, and Ki67 40%, HER2 negative. This is the Prosigna result. It's a luminal A cases with a 5% of probability of distant recurrence. Probably there is a misinterpretation of Ki67 evaluation. In this other example, 19% of estrogen, 70% progesterone, Ki67 under 50%, and the result it was luminal B. Also in this case, there was a problem in the classification, the determination of Ki67. Interesting also this one, in which we have 98% for estrogen, but 0 progesterone, with Ki67 80%, and the result, it was luminal A. Also in this case, probably there were some problem with immunohistochemistry with staining for progesterone. So some take-home message. Prosigna is based on PAM50, so it's possible to have information about molecular subtype. Is a second generation genomic prognostic gene signature, so incorporate clinical parameter. Is a decentralized and simple test. There is a demonstration for analytical reproducibility and about robust clinical validation. So thank you for attention.